Hi everyone, today we're going to make a model of polypeptide synthesis, which ultimately leads us to the formation of a protein. So before we begin with the model on the board, we'll just recap a few important components of the cell. So we have effectively a pancreas cell drawn here on the board, and we have the nucleus containing the DNA, which I have already indicated has become opened, okay, unwound and unzipped at a particular portion or a particular desired gene. Note that the nuclear membrane has small pores in it. However, the DNA molecule is a molecule that is too large and it can never leave the nucleus. We have the cytoplasm, and for simplicity I have drawn the cell with only one ribosome. However, in actual fact there could be thousands or millions of ribosomes throughout the cell, so this is a limitation of the model. Something else important to consider is in the nucleus, as well as the large DNA molecule, we also have individually floating free nucleotides of A, T, G, C and U. So we have adenine, guanine, cytosine, thymine, but also uracil, which we'll come back to later when we talk about RNA. So this video could be used in conjunction with this worksheet here outlining polypeptide synthesis, and specifically with this diagram here, which I've used as the basis for the model. So let's get started. Probably the two most important aspects of polypeptide synthesis that you need to understand are transcription and translation. And any model or HSC response about polypeptide synthesis would be seriously lacking if you failed to mention those terms. You will also need to be really critical in your usage of important biological terminology and refer to ribosomes, cytoplasm, codon, anticodon, transfer RNA, messenger RNA, amino acids, polypeptides, peptide bonds, proteins. It's also helpful if you can identify specific examples of proteins, because that helps you make your case. So just to recap, really important types of proteins that your body requires are large groups including enzymes, hormones, antibodies, and structural proteins, such as those found in the cell membrane. So the overall aim is to realise that genes found on DNA are expressed in the phenotype, that is the outward appearance of the organism, as proteins. Okay, so the coloured eye pigment in your iris, that's a protein. The tanning pigment called melanin in your skin, that's a protein. Hormones such as insulin are proteins, and all of these things have really important functional roles. So it is essential that genes code for the synthesis or production of polypeptides and ultimately proteins, so that multicellular eukaryotic organisms can function efficiently as a whole organism. We also need to remember at this point that we are talking about protein synthesis in what is called somatic cells. So these are normal body cells. And let's recall that in a human, every normal body cell, such as a nerve cell, such as a skin cell, such as a white blood cell, they each have 46 chromosomes. So each of those different types of cells in a multicellular organism is specialised. However, it does contain the full quota or complement of the 46 human chromosomes, occurring in 23 pairs. So we are talking about polypeptide synthesis in a normal body cell, and in our example, in a pancreas cell, that we are going to imitate the polypeptide and protein production of the hormone called insulin. Cells are specialised. So a pancreas cell does its job, a skin cell does its job, a nerve cell does its job, because particular genes on those 46 chromosomes are switched on. Only if a gene in a cell is switched on, and you can use that terminology in the HSC, only if the gene is switched on is it expressed. And what we mean by expressed is makes a protein. So if a gene is switched on, it will synthesize a polypeptide and protein, and it will be expressed, in our case, as the hormone insulin. The insulin is then available in this cell to do its job, which is regulate blood sugar. So let's recap. 46 chromosomes in any cell. For the cell to specialise, particular genes are switched on. For example, in our pancreas cell, we are going to suggest that this gene on our DNA double helix codes for insulin chain A, a particular polypeptide that will ultimately make insulin. So only certain genes are switched on. That means they're expressed as a protein. So let's get started. First step, and I will write the notes about this in a moment, the first step in polypeptide synthesis is the unwinding and the unzipping of our DNA double helix. So what I've shown here is already our DNA double helix unwound and unzipped. Now this only occurs at the desired gene. Okay, so the rest of the double helix is still wound up. We are only opening up the DNA at the desired section. And this is it here, the gene on this sense strand that codes for the polypeptide insulin chain A. This is catalyzed by an enzyme called RNA polymerase. So the next thing that happens, and what you should be noticing here, is that there are free nitrogenous base nucleotides in the nucleus. It is these free nucleotides that then come in and bond temporarily to our DNA gene that codes for our polypeptide protein of interest. So free nucleotides are going to come in and bind to the template strand of the DNA. So what we are doing here, what we are building here is actually a strand of mRNA, and that is messenger RNA. I've just prepared it earlier here. Very important things to note about the mRNA to distinguish it from DNA are that messenger RNA is much shorter, it's single-stranded, and it does not contain thymine. It contains the nitrogenous base uracil. So uracil, U, complementary matches to adenine A on the template gene in the DNA. Now the beauty of mRNA is that it is much smaller, so it can leave the nucleus via a nuclear pore. The DNA is a large molecule and it remains in the nucleus. This process of recoding the DNA gene into an mRNA strand is called transcription. The first very important process in this whole polypeptide synthesis process. So we have a complementary mRNA strand synthesized against our desired polypeptide gene. The mRNA leaves the nucleus, enters the cytoplasm, and is encountered by one of what is potentially thousands of ribosomes. At this point, we are going to call this translation. The ribosome is an organelle which you've been taught much earlier on in your schooling, is the site of protein synthesis. So now we're going to unpack that a little bit more. The mRNA is read by the ribosome and translated into a polypeptide chain, which is a series of specific amino acids bonded together by peptide bonds in a particular sequence. So what happens here is the ribosome reads the mRNA strand in triplet bases, and those triplet bases are referred to as codons. So one triplet base on an mRNA strand, our first is AGU, is called a codon. Our next codon is ACA, our third codon happens to to be UCA. Now, to initiate this process, there must be a start codon, which you can read more about in your textbook. Nevertheless, the process of translation is as such. In the cytoplasm, there is another RNA molecule, but this is called tRNA. This is transfer RNA. Each one of these molecules is 75 nucleotides long, and it is wound into the shape of almost a clover. Attached to one end of it happens to be a triplet nucleotide sequence. This triplet sequence is called an anticodon. Each anticodon on the tRNA codes for a particular amino acid, which are the building blocks of proteins. Each anticodon codes for a particular amino acid. So this tRNA molecule has an anticodon UGU, and in our particular example, I'm saying it codes for the light green amino acid. In reality, these amino acids have real names like arginine, aspartame, serine, which you don't necessarily need to remember, but understand that they're not colour-based. They actually have their own names. They're also in your textbook, I believe, on about page 125. So let's recap. mRNA strand is at the ribosome to be read. Each triplet on the mRNA is called a codon. tRNA is hanging out in the nucleus. It has an anticodon attached at one end, and that anticodon codes for a particular amino acid at the other end. So during translation, what happens is the transfer RNA delivers amino acids down to the ribosome, and the way, the sequence, the order in which they are delivered is based on the bonding between the codon and the anticodon. So we have AGU, transfer RNA also has U rather than T, so we're looking for a match UCA. And here it is. So transfer RNA travels through the cytoplasm, and the anticodon on our tRNA temporarily binds to the codon on our mRNA at the ribosome, all the while delivering this particular amino acid. The next codon on mRNA is ACA. So in this instance, we're looking for the match UGU. Again, we form a temporary bond between the mRNA codon and the tRNA anticodon, all the while delivering a specific amino acid. Now, as this process continues, between the amino acids, peptide bonds form. So I'm just drawing them in with a black texter. Ribosome, this is again poorly modelled here. The ribosome moves along the mRNA strand, but I can't do that because my ribosome is just a textured drawing. But in your model, it would be beneficial if you could somehow demonstrate that the ribosome is reading the mRNA strand by moving along it. However, let's bring our last transfer RNA strand in, and we have our temporary bond forming, our amino acid, specific amino acid delivered, peptide bond forming. So as this is all happening, the tRNA, who's already delivered its amino acid, splices off the mRNA and returns into the cytoplasm. So the tRNA has delivered its amino acid, it now splices or moves away from the mRNA and returns to the cytoplasm. These tRNA molecules will then go and pick up another amino acid that was specific to their particular anticodon. So if this was a blue sequence, it would pick up this one. If this were an orange sequence, it would pick up this one. But remember, the anticodon is specific in regard to which amino acid it actually picks up. So this process would continue as the ribosome reads the mRNA, and amino acids would continue to be added and formed along this chain until a stop codon of particular sequence was reached. And again, the particular order of that stop codon can be seen in your textbook on page 125, I believe. So what we have actually here is a polypeptide chain. It is important for students to realise, and this was a dot point in the old syllabus, it's important for students to distinguish between a polypeptide and a final overall functional protein. And the distinction is this: polypeptides make up proteins. So an overall protein could be one polypeptide chain that is folded up into a particular 3D arrangement to form a protein, or it could be numerous, that is, two or more separately produced polypeptide chains that need to find each other and wind up into a particular shape to form a particular protein. So, for example, I believe that the overall protein insulin that would be produced in a pancreas cell is actually formed by combining two separate polypeptide chains, insulin chain A and insulin chain B. So if this was the synthesis of our protein insulin, in my example, we're only halfway there because we have only synthesized one polypeptide chain. So winding back, this gene actually codes only for insulin polypeptide chain A. This cell would also need to have opened up at a different gene that coded for insulin chain B, and that polypeptide would need to have been formed at another ribosome so that both polypeptide chains could come together and wind up in a particular shape to form the overall protein called insulin. And only then have you formed the protein. So to be clear, lots of teachers, lots of textbooks, lots of students refer to this process as protein synthesis. More correctly, what I've demonstrated here should be referred to as polypeptide synthesis, because each time that occurs, you're only making one polypeptide chain. And some proteins are actually made of more polypeptide chains. So just be clear in your models that if your protein actually has more than one polypeptide chain required, you would need to recognise that in some way and demonstrate that different polypeptides are formed as a result of the expression of different genes on the DNA. Something else to consider, and this is how genetic disorders arise, is that if there is a mutation to your desired gene, that is any change to the base nitrogenous base sequence in your DNA, the mRNA will be transcribed incorrectly, the mRNA will then be translated incorrectly, and the polypeptide produced could either be incorrect or non-functional. Many genetic disorders arise in this way. Common examples of genetic disorders include type 1 diabetes, breast cancer, Tay-Sachs disease, Huntington's disease, cystic fibrosis, Marfan syndrome. There's a whole multitude of different genetic disorders that arise as a result of mutation. So in your model, it would also be really prudent to maybe identify a different type of mutation, whether it's a point mutation or a frame shift mutation, define those terms and make it clear in your model that a mutated gene will result in incorrectly transcribed mRNA, incorrectly translated mRNA, and ultimately an incorrect polypeptide, which could ultimately lead to the formation or non-formation of an incorrect protein. So just to recap, the process here whereby mRNA is synthesized is called transcription. And this is the recoding of DNA into messenger RNA. Messenger RNA leaves the nucleus, travels to a ribosome. The second very important process is called translation. This involves the bonding temporarily of an mRNA codon with a tRNA anticodon, the delivery of amino acids, and the formation of a polypeptide chain. So let's say mRNA plus tRNA and amino acid into polypeptide chain. Your textbook expresses these two processes much more eloquently, but this will give you the basis of polypeptide synthesis. Please remember, however, that synthesizing one polypeptide chain doesn't necessarily mean that you've produced your total protein yet. So it does need to be acknowledged that there might need to be further transcription and translation of a separate gene or genes to gain all of the polypeptide chains required to produce your overall protein. Okay, I think that's about it. Check your textbook for further details.